Peace be upon you. I am Sarkar. I'm going to explain this subject for today. Major histocompatibility complex. These are our learning objectives. I hope you get benefit from it. So basically, they are surface glycoproteins located on the surface of cells. Which cells? Before we go to that, there is two major classes of MHC molecules, class 1 and class 2. Class 1 is located on all nucleated body cells, but MHC class 2 is located on some special types of cell with antigen presentation, and these are macrophages, dendritic cells, and B cells. We will go to details in next slides, but what they simply do, they express antigens in this pocket here and also here to some other cells. For example, this is MHC class 1, express or displays antigens to immune surveillance cells, and the immunocyte, for example, natural killer cells, will check whether the cell is healthy or not. And this is expressed on antigen presenting cells, especially when they capture extracellular antigens, and they go to lymph nodes and display them for lymph node resident cells. I mean T cells and B cells. If we go to a historical perspective, before the studies start, it was known that tissue transplantation between non-identical twins or non-identical individuals was rejected. Otherwise, the same tissue was going to be accepted between identical twins. So they thought, scientists thought, this has a genetic base. So what they did, they produced some kind of mice which to be genetically homologous at every genetic locus. This is one kind. So these are called syngenic. And they produced another kind which is called congenic, which have genetical difference in only one genetic locus, and which was MHC locus, which was located on chromosome number 17. So what they did, they grafted tissue between these mice, and what they saw, tissue graft between the same strain of mice, I mean syngenic mice, was accepted, but tissues which grafted with congenic mice or between congenic mice simply was rejected. Later on, they found the genetic locus responsible for this rejection was located on chromosome number 17. And before this, it was known that this locus encodes a blood antigen called antigen 2 in mice. So hence, it's called H2 complex in mice, or histocompatibility 2 complex in mice. Later on in humans, also it was known that people who have received multiple blood transfusions and tissue grafts from other individuals or different or non-identical individuals, simply they had antibodies to some antigens which were located on leukocytes. So it took the name of human leukocyte antigen because the antigens which were responsible for the rejection were located on human white BCs. So they called it human leukocyte antigens. At the same time, when they found that the MHC locus was the major determinant of tissue rejection or acceptance in mice, later on in subsequent studies, they revealed that the proteins encoded by these genetic loci between humans and mice are very similar in structure, so they got a strong homology. So by this way, they reveal that there is a combination in both, or there is a relationship between tissue or the fate of tissue when they are grafted with these genetic loci, which were HLA in humans and H2 complex in mice. Later on, scientists reasoned that are these genetic loci only responsible for the fate of tissue, or they have other functions. So what they found was that they are very fundamental for immune response, so hence called immune response genes. And this is revealed from some experimental studies, which we will talk about in later of this subject. Okay, so before we go on, we should know why these MHC molecules are important, because as you know, we have T cells and B cells, which are a very important part of immune system. T cells can't recognize soluble antigens unless they are expressed. They are at first they should be engulfed and processed, then expressed on special proteins which are called MHC proteins. So as you see, for example, this is an antigen presenting cell, MHC2. This yellow is the antigen which is held on this red. This red is MHC class 2 to display it for this, as you see, immature CD4 T cell. This is a T cell or T helper to be recognized by T cell receptor. Then if it requires the activation, it will do. This is the same for CD8 T cell or cytotoxic T cells, but the only difference is that the antigen should be displayed on MHC class 1. Even for B cells, it is right that B cells can recognize some antigens which are called T independent. For example, as you see, it has made cross-link with B cell receptor. This particle, it could be a bacteria, and this one, C3D. But this is not very common. The common way, it is right that B cell can recognize antigens for itself, but it can get activated without T cell help for most type of antigens, which are called T dependent. So even B cells require the T cell help for activation or for fully activation. As you see, this an antigen presenting cell hold peptide on MHC class 2. This T cell receptor, this is a T helper. For example, if we suppose that this T cell is activated, then it gives the help for B cells, which has recognized the same antigen for activation, and this leads to activation. So this is why it is important. There is another concept, is MHC restriction, is that this T cell receptor can recognize antigens displayed on MHC molecules from the same individual. I mean, from an individual who have inherited the same genetic components. Because, for example, if you derive these T cells and or you have stimulated in vitro for the same antigen, and in another study you activate this antigen presenting cell, but you derive them from another individuals who is genetically different with you, simply these T cells won't get activated. We will study in next slides, but this was only for basic information. About the structure, so we have class 1 and class 2 MHC molecules, and there is a difference between them. Let's see what is as we talked before. Class 1 is expressed in some kind of cells, and this class 2 is expressed on or some other kind of cell or some specialized kind of cells. And now we jump to study the structural basis of MHC molecules. As we see, MHC class 1 is composed of alpha chain and beta 2 microglobulin. The alpha chain is composed of 3 domains, which are about 45 kilodalton and a 12 kilodalton beta 2 microglobulin. This is the external domains, and about the transmembrane domain is composed about 25 amino acids and 30 amino acids tail, which is located in the cytoplasm. The external structure is about 90 amino acids. This is encoded by MHC locus, but not this one, not MHC microglobulin. They are non-covalently bound to each other. Despite that, they have a homology between them, so they are structurally near. Despite that, they are not coded on the same genes. The domains which interact with the antigen are alpha 1 and alpha 2, which are also called membrane distal domains. The antigen locates in here or sits in this groove, which is formed by beta sheets here, which we will study later. These domains are the membrane proximal domains. About MHC class 2, actually the difference is that there is no beta 2 microglobulin in the membrane. Sorry, I did a mistake. Alpha 1 and alpha 2 are membrane distal, these are membrane proximal. There is no beta 2 microglobulin in membrane proximal domains, but it is basically composed of 2 chains, alpha chain and beta chain. The alpha 1 and beta 1 are located or are the membrane distal domains, and the alpha 2 and beta 2 are membrane proximal domains, and the remaining is the same with MHC class 1 molecule. However, the alpha chain is about 33 kilodaltons and the beta chain is about 28 kilodaltons. So the alpha chain it looks to be larger. We talked about the antigen interacting area is also a groove formed by combination of these 2 chains, which also are non-covalently bound to each other. Other things we will study here about the this is also class 1, but in another different picture. The beta 2 and alpha 3 domains are simply composed of beta sheets, many beta sheets. The important part is the the antigen interacting domains, which are formed by alpha 1 and alpha 2. As we see, the alpha helices, many alpha helices, as we see here at the sides, and the platform at the floor of the peptide binding domain is formed by beta sheets. As we see here, there is one characteristic of MHC class 1 which have some conserved residues at the end of the MHC class 1 or at the end of this peptide binding groove. There is some conserved residues here and here which interact with the antigen, which have some kind of special amino acids usually they are hydrophobic, which act as an to anchor the peptide in this groove. So the peptide basically it is bound to these two areas, make hydrogen bonds here and here, and the remaining part makes a bulge which interacts with the T cell receptor. So usually the antigens which react with class 1 MHC are small, should be small, because they should accommodate this area. Usually about 8 to 10 amino acids, but about MHC class 2, they have very similar structure except for difference in here. I mean the domains which form the peptide binding groove is beta 1 domain and alpha 1 domain at each side. Again, alpha helices at the side, but there is no conserved residues at the peptide binding groove. So the hydrogen bond between the peptide and the amino acids located at the binding groove is formed throughout the area. So it's not only located at the ends, the amino end and the carboxylic ends. No, here the peptide interacts with this area, or the hydrogen bonds are formed throughout the area. So it could be here, it could be here, some could be here, and some could be here. So this gives us the advantage. MHC class 2 molecules can interact with larger peptides, which can be about more than 10, or it could be sometimes 20. Even some studies say it could reach 30 amino acids. We'll study more details here. As I told you, these act as an anchor. For example, some amino acids here. These are the major amino acids that do this function to anchor the peptide, and the center is usually variable region which interact with the with the T cell receptor. These are the antigens, sorry, amino acids which act as an anchor we just talked about. About the MHC class 2 and peptide interaction, as I told you 2 slides ago, it is like an open end for MHC class 2, so there is no conserved residues, and the peptides also the peptides which interact with class 2 MHC molecules don't have conserved residues, so the residues which are complementary to the peptide binding groove are located throughout the peptide and make a constant elevation on the floor, which is formed by hydrogen bonds. But what is important here, the core of about 13 amino acids, or 13 to 18 amino acids would interact with T cell receptor, are the most important residues in the peptides. So the larger peptides can go extend beyond the ends. Before we go on, there is a characteristics of MHC molecules is that there is a concept called promiscuity. So MHC molecules are promiscuous. So they can bind for example, one MHC molecule can bind or can recognize several distinct antigens. So also the antigens can bind many different MHC molecules, and later on we will talk why this promiscuity is important. Despite having many similarities, also there is some differences, as we will summarize it here. The alpha 1 and alpha 2 for peptide binding group for class 1, but alpha 1 and beta 1. Closed nature of peptide binding groove, closed at both ends. and open at both ends. General size of amino or general size of bound peptide is about 8 to 10 amino acids. Here 13 to 18 amino acids. Anchor residues at both ends, conserved residues distribute along the length of the peptide. Extend structure, okay, at both ends, but the middle arches up, so there is no bulge here, but the peptide is held at a constant elevation above the floor of MHC. Now we will talk about the MHC loci or MHC locus. These genes are located on chromosome number 17 in mice and chromosome number 6 in humans. As it is depicted in this diagram, there is class 1, class 2 genes and class 3, and we will talk about differences between these. Why it is written here HLA complex? Because I told you before, at first it was discovered as human leukocyte antigen. So the genes that encode class 1 proteins are HLA-A, B, and C. What they encode class 1 MHC molecule, which is a glycoprotein expressed on the surface of nearly all nucleated body cells, and the major function is to express or display antigens to, for example, CD8 T cells, not CD4. About the class 2, the genes, the major genes here are DP, DQ, DR. So because class 2 is composed of 2 chains, they should encode both alpha and beta chains. How about class 3? Actually, it encodes some very important protein which have important functions in immune system, but they have nothing to do with major histocompatibility complex. But they have other functions. Also, there is many these basically, as we see, for example, these are 3 genes, and this is depicted here in a different way, but they are composed of many different types of alleles. But simply we don't inherit all alleles, which we will talk later about. Let's see what are these black colored genes. For example, DO and DM. So what we just talked about class 1 and class 2 are called classical MHC molecules. So why? Because they simply do their function by displaying peptides to CD8 and CD4 T cells. But there is also non-classical MHC molecules. For example, there is a HLA-G. Studies say it has a very important function with discrimination of self and non-self. For example, they present on fetal cells at the maternal fetal interface and are credited with inhibiting rejection by maternal CD8 T cells, by protecting fetus from identification as foreign, which may harbor some paternally derived antigens. So this is not a classical function. Also for MHC class 2, we have, as I said, we have DP, DQ, DR, which encode both chains, but there is also non-classical MHC genes. For example, designed as DM and DO. Also, they have nothing to do with the classical function, but for example, what they do, both HLA-DM facilitates the loading of antigenic peptides onto class 2 MHC molecules, and class 2 DO molecule, which are expressed only in the thymus and on mature B cells, have been shown to serve as regulator of class 2 antigen processing. So there is a class 3, let's see. The products of class 3 are complement proteins, for example, C2 and C4. Factor B is also involved in complement system and tumor necrosis factor and lymphotoxin A. So as we see, also these are not classical MHC, even don't encode anything related to MHC molecules, but studies say, despite differences from class 1, class 2 genes, the linked class 3 genes cluster is conserved in all species with an MHC region. So why? This suggests there is a similar evolutionary pressure have come to bear this cluster of genes because they also have very important functions in the immune system, and the polymorphism or mutation in these genes may lead to very serious disease. For example, Crohn's disease, autoimmunity, arthritis, and so on, especially mutations related to tumor necrosis factor. So this was about the genetic locus or the locus of HLA in humans. About the exon and intron arrangement, actually it is very simple, but for class 1, as we see, it is a single, sorry, not, it's composed of a heavy chain and a light chain. What is important here, they are both for MHC class 1 and class 2, they are flanked by a promoter which enhance to or facilitate the insertion of the alpha chain into, not actually the alpha chain because it is located on the alpha and the beta. It facilitates to the insertion of the products of transcription through endoplasmic reticulum, which is then removed proteolytically. What is important? So I think it is very clear. About pattern of inheritance and expression. This is very important subject and take notes. We said that before there is particular genes which encode different parts of the MHC molecules, but there is no single gene to do this, but they come with different or alternative forms of each gene. So studies say the genes that reside within MHC region are highly polymorphic. So what does that mean? The gene is the same, but in different forms. What we say, for example, we studied or we know that in Mendelian traits, for example, there is a gene to code the color of some kind of flower. There is two genes, the red one and the white. For the function, they are the same, both code for color, but in the form, in the shape, they are different because one encodes a red color, one encodes a white color. So that's how I mean the genes are the same in function but different in form. So they are polymorphic. And what we call this is alleles. So they come with multiple alleles for each gene. What is also important is that they lie very close together. So when they are inherited from an individual to another, they are transported as a set. So what we call is the set of linked alleles is referred to haplotype. So don't forget this because we need to remember in next slides. But before we go, as we see in this peak, this mating between humans, not mice. For example, this is haplotype A and B, and C and D for the women. And why humans are different, or they have different haplotypes, they are not syngenic as mice, because humans are outbreed population. What I mean, they mate with different individuals who possess different set of alleles. So basically most of humans are heterozygous except for identical twins. This has led to a huge diversity in human population because human is a species. Oppositely, mice mate with each other, so you could find some mice which are homozygous or syngenic to have the same kind of alleles, and this is actually for the function of immune system. This is not good. So when they mate, they produce different combination. For example, A with C, A with D, B with C, B with D, but there is a very small percentage or possibility for happening of recombination during meiotic divisions. So the possibility is only 0.5%, but it was enough to make this huge diversity throughout the whole human population. There is one point I want to declare is that why this huge diversity should exist for MHC genes, because once these alleles are transferred from, for example, the mother and the father to an individual, these encode proteins which are constant and they won't change again. What I mean, for example, in B cell and T cell receptor, they continuously do some kind of mutations to produce a very specific type of T cell and B cell receptor which can only bind to a single antigen. They can't bind to, or they can't recognize other type of antigens. There is no promiscuity in them because they have the ability to do that, to recognize very specifically for an antigen. This kind of specificity does not exist for MHC genes because, for example, somatic hypermutation, or some other mutational process aid to diversify or maximize the diversity of T cell and B cell receptor, which happen during their development and even after their development. This basically doesn't happen for MHC genes. So basically what an individual inherits is the same until he dies. So this is why promiscuity should happen in MHC genes. So they should recognize for example, different peptides which are different in structure. This is to maximize the possibility of recognizing even unchallenged antigens or the antigens which have never been seen. This is why promiscuity should happen in MHC genes, which doesn't exist, or the high specificity of T cell and B cell receptor doesn't exist in MHC genes. The MHC genes are codominantly expressed. So what do I mean is that, for example, they are not as Mendelian traits. For example, when you inherit two alleles for the same gene, one is dominant, which may make the recessive one to disappear. Here is not, but both are expressed equally as we see here or as we see here. These are all genes for class 1 and class 2. You see all are expressed. None of the genes are suppressed, or none of the alleles are suppressed. This is codominant expression. As we see here, for example, mating between a mice with this haplotype BB, B small, B small, K small, K small, they produce BK haplotype for F1 progeny. So skin transplantation between inbreed mouse strains with the same or different MHC haplotype. For example, when you do tissue transplantation between inbreed mice or syngenic mice, these have the same MHC haplotypes. Simply, it is accepted because they have the same MHC loci. What about this one? This have K small, K small. This is a different haplotype. Simply, it's not accepted because the MHC molecules here are different. However, it can be accepted by this individual because it possesses the B haplotype, B small. And what if this one, the F1 progeny, gives tissue to its parents? None of the parents will accept any tissue from the progeny. Why? Because it possesses a haplotype from, for example, the yellow one that you see, the K, and the K is rejected when you inject it or when you graft it in the BB's haplotype. What else? Diversity at the level of both individual and species levels. So as we know, a particular MHC molecule can bind many different peptides. This is what's called promiscuity, which gives the host advantage in responding to pathogens. Rather than relying on just one gene for this task, MHC region has evolved to include multiple genetic loci encoding proteins with the same function. Despite that, we talked about, but why I talk about again, it's because it is very important to note. We simply humans, in general population, there is hundreds of different allelic variants for class 1 and class 2, but any human can only inherit 6 different class 1 molecules or allelic variants for class 1 molecule and 12 for class 2. Why 12? Because they are composed of 2 chains. When we inherit 6 alleles from mother and 6 from the father, different combination of the alpha and beta chain lead to another diversification process of the MHC class 2. But for class 1, what you inherit is directly encoded. For example, as we see this from the mother, it's directly encoded. This is from the father, it's directly encoded. So there is no such a different different combination between the chains. MHC regions are said to be polygenic because it contains multiple genes. Okay, we talked about this. Why this diversity is important? It is important actually because each human should try or to maximize the antigen recognition capacity of this MHC molecule. So this what lead to some people in a species, for example in humans, are resistant for some kind of disease, and otherwise some individuals are susceptible. Why? It could be attributed to many things, but one of these are different peptides can be recognized by different MHC molecules encoded by different allelic variants of MHC. For example, there could be some alleles more important for immune system or immune recognition or T cell and B cells to target them than some other kind of antigens. This is one of the reasons why diversity is important, because body tries to maximize the diversity so as to recognize a huge number of distinct peptides, uh, so as to come over any, for example, an infection or an infectious agent, for example, a virus, a bacteria. Let's show you an interesting thing before we go. Oh, yes, before we go on. In some studies, it's been revealed that single nucleated cells express about 10 power 5 copies of each of these unique class 1 molecules. Okay, this unit, for example, this allele can be expressed about 100,000 copies. Okay, one allelic type. In a critical study of peptide binding by MHC molecules, peptides bound by two different class 1 MHC proteins, so it means two different or two allelic variants of MHC class 1, were released chemically and analyzed by HPLC, high performance liquid chromatography mass spectrometry. What they revealed, it is so interesting, just pay attention. More than 2,000 distinct peptides were found among the peptide ligands released from these two MHC molecules. Suppose that these two MHC molecules have been tested in this study, and they have seen that 2,000 distinct peptides, distinct means different in structure, have been released. So it means that from two allelic variants, they have the capacity to recognize 2,000 distinct peptides.